Hello and welcome, grade 12 mindsetters, to today's exam school. And today's exam school is going to be on life sciences. I'm here with your favorite bio teacher, Aslam. How are you doing?
Good, Lloyd, and yourself?
Yeah, I'm all good. I hope you guys are keeping well. I hope you guys, your guys' exams are going really well. And I hope you guys are ready for this lesson, for the exam on life sciences. But without wasting any more time, let's get ahead of the lesson, shall we?
Thank you, Lloyd. Welcome, Mindsetters Grade 12s. Can't believe where we are at this time. We are there on the eve of your paper one. Yes, that's right. You write paper one tomorrow. I said here we're going to revise all questions on paper one. I see there's lots of confusion. The newspapers are talking about it, that learners have on their timetable. They've written their paper one structures, and paper two they've written environmental. Remember, that is incorrect information. Unfortunately, that slipped through the system. However, you that are watching Mindset from the beginning of the year, you have absolutely no excuse, because from the beginning of the year, we have been outlining to you what is paper one and what is paper two. Your teachers probably did the same thing, and I have been talking about the exam guideline continuously. So you must remember what is paper one. Let's just talk about paper one today. We're not going to talk about paper two, so you don't have any confusion. First of all, in paper one, yeah. I'm going to remind myself, it's meiosis for 11 marks. It's nervous coordination. Now, I'm writing nervous coordination. Your exam guideline talks about responses in humans. That includes the brain, the spinal cord, reflex arc, eye, ear. That's what's included there. That is 40 marks plus minus. This is a guideline, remember that. Then we have the hormones in humans. That means the endocrine system, that is 15 marks. Then we have homeostasis. I may be out here and there with the 1 or 2 marks, forgive me for that, but more or less, I think I'm spot on. Homeostasis, then we have reproduction in vertebrates, that means all those strategies for 6 marks, and we have reproduction in humans, a whopping 41 marks. Hormones, reproduction, homeostasis. Then we have environment, which is 25 marks, and plant hormones. About 12 marks, I think, as well. More or less, that should add up to about 150. Like I said, I may be off, rounding off here and there, but these are the sections. Remember, meiosis is in paper 1 and 2. The same meiosis is in paper 1, paper 2. So when you learn it once, you score twice. So those are the things that you need to know. Let's not waste time and go straight into the actual lesson. The first question, and this is on the page, yes, Lloyd?
Yeah.
The challenge question.
Yeah, guys, all the stuff from today's show, it can be found on our Facebook page. So this paper, as well as all the things we're going over today.
Good, excellent. Thanks, Lloyd. Okay, so the question says, study the following diagram representing a process in humans and answer the following. So they have something drawn there with some tadpole-looking structures that are moving towards them, and something is happening on the inside there as well. They got some labels A, B, and C. Identify the process taking place. Where in the human female body does the above process occur? The minute they say human female body, that already tells you that this section is from reproduction. Nowhere else will we distinguish between the male and the female besides reproduction in this paper. Assume structure B had 24 chromosomes instead of 23. Explain how structure B could obtain 24 chromosomes. How come not 23, it's got 24? And how many chromosomes will structure C have if structure A was normal? So those are the things you have to look at. I'm not going to waste your time because you can read that. Let's move on with the questions. We start with environment. Remember, environment is 25 marks in this paper, and it's a fairly easy section because you've done it in grade 11 and again in grade 12. The same section, it has not changed. So you should know how to work it. And 9 out of 10, you get questions that deal with investigation type or data, that is their data response. So let's look at this one. Access to clean water in South Africa varies considerably. Immediately they're telling you clean water, your head must go that this is dealing with water quality. Okay, so that's the section that we're dealing with. Depending on the source of water, a group of pupils conducted investigation to determine the level of satisfaction of people who obtain water from different sources, 4 different sources. One is boreholes, farm dam, river and pipe water, that means the tap. That means you and I opening the tap and the municipal water that we are having. The second column, I should say, that's the first column, second column, third column. The first column tells you the percentage of people that are satisfied. 73% were satisfied with borehole, 68 were satisfied with farm dam, 54 and 79. I don't have to read the other one because that will be the difference. 100 minus 73 will give you that one. Question says, draw a bar graph to represent the results of this investigation. So all we are doing here, guys, and you don't have to study for this, you just need to know the skill of drawing a graph. You're taking this information and you want to draw a graph from this information. Remember, the two variables are water source, and these two are the other variable. They're either satisfied or dissatisfied. The independent variable, the one that the investigator can change, is the water source. You can't change whether people are satisfied or not. That will come from what is happening here. So that's your independent variable. Your dependent variables are these two. The levels of satisfaction, I would call it. So that's your two variables. And those two variables must be captured in your heading. So when you're drawing your bar graph, that's what you're going to have. So you're going to say graph showing. In fact, in reality, even this graph showing you can take out now. You can just start here by the level of satisfaction or dissatisfaction with, that's a one variable. That is the dependent variable, with water quality from 4 water sources. So that's your heading. For having that whole heading, you're going to get only 1 mark. But you must have both variables. That moves on to the y-axis and the x-axis. So here we're going to talk about source. Here we have percent people. So percent is already given. You don't have to put a unit. And source can't have a unit. Generally, in a bar graph, you won't have a unit as such. But if you have to, you'll put the unit next to this. And this is how you draw it. Now, I'm not going to go through the graph. I'm only going to tell you the issues that you have there. First of all, if you have this situation, you're going to have a key. You must show the key. Right? Here you have your headings for the different ones. Notice that the bars are equal width. Each bar is the same width as the one next to it. And each group is separated from the other by an equal amount of space as well. Notice, because we're talking about satisfied and dissatisfied, we can keep these two bars together, even though this is a bar graph. Remember, in a bar graph, the bars must be separated. But we have groups of bars, so the groups will stay together. They will be separated from the next one and the next one and the next one. If you do not want to have a key, you can simply write in the bars. For example, this is borehole, this whole thing is borehole is there, satisfied, dissatisfied. You're going to write it in full, obviously, so that the person who's reading your graph must be able to see clearly what's happening there. But that is how you present it. Remember, you can also be tested on a histogram where all the bars will be together, or a line graph where you're going to put points and you're going to join those points. Line graph, and you can be asked to draw a pie chart with different sectors. Remember, to draw a pie chart, you need a protractor, you need a compass, and you need a nice sharp pencil, because you cannot estimate those angles. You've got to work it out. And when you do a pie chart, you must show the calculation. How did you get to this angle or that angle or that angle or that angle? Remember, when we mark this, we put a transparency over your pie chart and we move it around to measure your angles. Generally, you are only allowed the error of 1 pencil width. So you can't guesstimate and just draw your own things there. And your circle can't be like mine's here, because for a start, my circle is not a circle. It's skewed up everywhere. So if you're using a compass, then you can do that properly. Make sure you have the right equipment. The next question says, borehole water is obtained from an aquifer. Obviously, you need to know your work. If you don't know what an aquifer is, you're screwed. Okay? Briefly explain why the water from this source, which source? From the borehole, may be of similar quality to piped water. How come the borehole water can be, according to this, look at the satisfaction levels also. Well, we can look at it in our graph. Here's borehole, just above 70% pipe water, just below 80%. So they're in the same range. People are equally happy there. And the reason is obviously the water filters through the sand or the rock. Remember, an aquifer is a rock that is porous. It has pores. It has tiny holes in it. So those holes or pores act as filters. So they're going to purify or clean the water during the filtration process, just like the tap water goes through many filtration processes as well. So the question really is testing whether you understand what an aquifer is. The aquifer is the rock from where we're going to get water. Why? Because this rock is porous and water seeps in through the pores, and we then extract that water from there. Which water source provides water which participants were least satisfied with? I mean, if you don't understand English, then you are in a problem situation here. Remember, percent satisfied is shown with the lined areas there. You got to look for the lowest one here. And Lloyd, which is the lowest one here? 1, 2, 3, or 4?
4.
The lowest of these ones I'm talking
Oh,
about.
those ones.
Yeah.
Oh, 3, yeah.
3. Thank you, Lloyd. Otherwise, we would have given you 0 there. Okay. Okay.
Sorry.
So this one here, and that is the river water. So they are least satisfied. And people, you didn't even have to look at your graph. You can look at the table. Here's the numbers. 54% is the lowest number in terms of satisfaction. So therefore, it's river water there. So the people are dissatisfied with river water, and it's for obvious reasons. And suggest one way in which people who receive water from this source, which source? River, would treat the water themselves. The most obvious answer would be, I would first give this answer. That's the easiest one. Boil the water before you're drinking it. In addition to this, you can treat the water with jig or bleach. Remember, this is for cholera and other diseases that we can deal with. Filter water through clean sand. You can take sand, put the water through, let the water seep through, and that will filter the thing as well. Or use what they call now, we get purification tablets that we could put into the water that will purify it. But the first and the easiest of them all, I would put this right on top. Boil the water before you drink it, because boiling will kill off any of the germs or bacteria that is in the water, disease-causing bacteria. Briefly explain how each of the following influences the water quality. Okay? Water quality, thermal pollution. Thermal coming from the word temperature. And thermal pollution means we are increasing the temperature of water somehow. And this is obviously because of releasing water from factories and mines. Remember, these factories and mines have large machines. These machines need to be kept cool. The thing that keeps them cool, just like how your motor car engine keeps cool with water, these machines are kept cool with water. And these are larger than your engine. That water gets hot. That water has to go away to be replaced with cold water. That water is released into the water. It takes the temperature of the water up. The problem with that is higher temperatures affects the amount of oxygen in the water. The higher the temperature, the lower the oxygen content. And that's where we get our answer from here. So high temperature or heat reduces the oxygen in the water. Organisms in the water will die because of lack of oxygen. Now what will happen? The bacteria act on the dead organic matter and use up even more oxygen, because the decomposition process will take place, and this is going to attract the bacteria. The more oxygen is going to be depleted. Well, oxygen will be depleted even further, rather put it that way. Excess fertilizer. Remember now what we're saying. The farmer is spraying or putting fertilizer on his land. If he uses too much of that, what can happen to it? It can wash away into the river. What is fertilizer? It's excess nutrients. That means now the plants in the water, they're going to get these nutrients. If they get these nutrients, they're going to grow much more. They'll form a blanket on the water called algal bloom. Less light to the bottom. Those organisms at the bottom of the river or stream or dam will die because they're not getting sunlight. And the organisms that depend on them will also die. Now again, they're going to attract bacteria. Bacteria deplete the oxygen further. Again, this is going to kill more animals and plants. So remember, animals and plants dying occurs twice in this answer. One is, firstly, because they're not getting light. Secondly, after the first set of organisms die, the second set will die because of the decomposition process. So fertilizers firstly. And again, please watch the step-by-step answer. You can't just say eutrophication and jump to that. You start by saying that the fertilizers get into the rivers when it rains heavily, first point. Second point, this provides nutrients for rapid growth of algae, all right? Also known as algal bloom. Thirdly, it covers the surface, what? The alga, and blocks out the sun. Fourthly, the water plants cannot get sunlight, and they die. If they die, the animals depending on them may also die. Bacteria decompose those things and use up more oxygen. So 6, look as I said, other organisms now die of suffocation. So the dying of organisms take place at 2 levels. One is the first level because they're not getting sunlight. The second level because oxygen depletion is taking place. With that, Lloyd.
Yeah, guys, so before we go on to our ad break, remember there's been one thing we've been talking about the whole of exam score, and I hope you guys are excited to. This Sunday, we announced the winner of that brand new Chevrolet Spark L, and it could be you guys. Remember, tune in on Sunday evening at 5 p.m. and you could be the winner. Any of you who have bought your K53 book or bought the app, you have been entered in. If you have gone onto our page and entered into all your details, it could be you. Guys, if you still want to buy and still want to enter that, we are still giving away 3 more cars for the next 4 rounds. All you have to do is get yourself this K53 book or buy the app, enter on our Facebook page, and it could be you. But guys, don't go well. We'll be right back after the break. Welcome back, Matric Mindsetters for Life Sciences. Guys, remember the show is sponsored by the MTN SA Foundation and is proudly endorsed by the Department of Basic Education. So guys, before we carry on, there's been a lot of questions on the essay question.
What's the actual question there, Claude?
Sid, could you give me a possible essay question, please?
Guys, you can't ask us that type of question, right? You can't ask us what's possible essay. We are not magicians. We don't see the papers, etc. So we can't give you that information. Remember, one thing is guaranteed. What is the guarantee? There is an essay in the paper. Second thing is guarantee. This essay is based on the syllabus. So what conclusion you can make from that? Learn your work so you can answer any essay.
Yeah, I doubt they're going to ask you a question that you don't know.
They can ask essay from any of the topics. Don't be fooled by seeing that endocrine, for example, is 11 marks or plant hormones is for 11 marks, so therefore it doesn't make 20, it can't be. What's stopping from anybody from putting 2 sections together and asking an essay? Look at 2013, look at 2012, look at 2011, and look at the type. Important is that there will be an essay. The essay is always going to be 20 marks. The essay will always come from the syllabus. Know your work, the essay becomes easy. So keep that in mind and understand and try to learn the skill of writing an essay. And the basic skill is look for the verb. Whatever comes after the verb is what you're going to do. We're also going to look at some essays just for the skill. Remember, it doesn't mean the essays we have here are possible essays. There's no such a thing. Please. Okay? Let's move on. Thanks for that, Lloyd. We'll take some other questions a bit later. So we're moving on with environment still. The table shows the amount of solid waste generated in a town that was disposed of in landfill sites over a period of 6 years. They're giving you the figures 1999 ending in 2004 and the amounts to 255 going to 428. Data response again. Describe the general trend. So we're killing 2 birds with 1 stone here. We're talking about solid waste. At the same time, we're learning how to answer data response question. That is what you must take home. Take home the skill so that you can answer any other data response. Describe the general trend. General trend means what is happening in this graph from 1999 to 2004. And you can see higher, 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 higher. So the general trend is that the total solid waste is increasing from year to year, or the total solid waste is increasing over time. That's what we need to give there. The amount of solid waste by the town is increasing from 1990 to 2004. One is produced is increasing, and for giving the period, or by saying over time, or year by year, you're going to get the second mark. It's better to give the time period because then you are referring to the data. In which year was the solid waste generated the highest? Now that's a giveaway. If it was increasing from 1999 to 2004, obvious it will be 2004. That's, again, you don't have to study for this, people. You have to look at this information and answer based on this information. This is the skill you need to take in this particular question. List 2 strategies that can be used. List 2 strategies that can be used, and the verb is list. So you don't have to explain it, you have to list it. That could be used by municipalities to slow that out. And I'm not going to waste time with that one, that's straightforward. First of all, remember, it's the municipality. It's not about the individual. So the municipality must encourage people to recycle. One, you're not explaining what recycle means. Educate people about landfills and make them aware about landfills. Fine people who dump. So in other words, we must have legislation, put laws in place to fine people who waste or dump waste incorrectly. Teach people how to build their own compost heaps. So this is now an innovative way. Instead of going the harsh way, you're going to another way. Also by legislation, you can also say, instead of fining, you can give people incentives to reduce waste. In other words, if the people give you less waste every week when they're collecting the garbage, then you can give them incentives for that. Okay? One problem associated with landfills is leaching. Big word for small learners. Explain what is meant by this term. Straightforward again. Leaching means to leak out. So what's happening in this particular case, poisonous or harmful substances in the landfill dissolve in water and filter through the landfill into the groundwater. And this is going to cause problem with the groundwater. It can also then go into the soil, and that can also cause problems for plants growing in there. But more importantly, it's affecting the water table. Are we good? Good. Let's move on. Okay, so that's a little bit of environment. We put that first because we did that last in the past, so we put that first so that you get some. Remember, environment, you still need to know atmosphere and climate change. You need to know food security. You need to know water quality, water availability. You need to know about deforestation and the rest of it that goes with all that stories there, and solid waste. Those are the areas that you need to concentrate on. Okay, the following diagrams are representation of a real life situation, right? It's taken from not from the KZN paper, it's taken from another paper. Study the diagrams and answer the questions. So there's a real life situation. Here's a person reading a book in one area there. Another one is looking at a traffic light in the distance, far away. We can't see very much the shapes of the different lenses, but you should know these are the clues given there. The first one says, identify the eye mechanism represented by these diagrams. What about the eye are we discussing here? Obvious, we are dealing with accommodation, seeing near and far, less than 6 meters, more than 6 meters. Okay? Briefly describe what changes took place in the eye from diagram A to diagram B. From diagram A, there, to diagram B. That was far, this is near. So actually this question is asking you, describe near vision. Where do we start with this? I already taught this section when we spoke about logic, and I started at the back. Today we're going to start in the front, or rather at the top. So the ciliary muscles contract, and I always put this in here to the explanation so it makes logical sense. Ciliary muscles contract towards the lens. So figure what I'm saying. The ciliary muscles are moving inwards towards the lens. And if they are moving inwards towards the lens, they release tension on the suspensory ligaments. And if they release tension on the suspensory ligaments become slack. And if they become slack, the lens becomes more convex. And the lens becomes more convex, the light is refracted more. So refraction increases. And focal length, I'm putting this is not for marks, just to make sense. decreases, and a clear image falls on the retina. Whether you're seeing near or far, this last sentence must always be there. A clear image must fall on the retina. Now, what happens if we ask for far vision from B to A? Easy. If you know this, the other one is easy. We're going to change that to relax. The ciliary muscles will relax. Obviously now away from the lens. So you're going to imagine that away from the lens. If they're moving away from the lens, there's going to be more tension on the suspensory ligaments. And if there's more tension on the suspensory ligaments, they become taut. Remember, they are ligaments, so therefore we cannot use the word contract and relax. We use the words slack and taut. Taut means tight, slack means loose, okay? Now, because of that, if they become taut, that means they're pulling as well. And if they're pulling, the lens becomes flatter. Or, if you don't want to use that, you say less convex. And if it becomes less convex, then refraction will decrease, focal length will increase, and what do you have? This sentence stays the same. Now, guys, you only have to learn one because the other one is the opposite. You have to learn this. This is easy stuff to learn. It's easy marks. If they ask a question like that with diagrams, it's easy to answer that question. For a start, the diagram is staring you in the face. You are simply saying what you're seeing in that diagram. Okay? So that's what you do for accommodation. Okay? The diagram below represents some eye disorders. Study them carefully and answer the questions that follow. Okay? This one is showing you nearsighted vision and far-sighted vision. Explain why a far-sighted person's vision is distorted. This is straightforward from the book. First of all, a far-sighted person, the image is falling behind the retina, so he can't see it. It must be on the retina. It's not inside, it's on top. You know that one? We're not going to say the brand name here, but you know what I'm talking about. This is caused why? Because the eyeball may be too short. This is genetic. So the image is falling behind the retina. Or the lens cannot become round enough during accommodation. This may be genetic. You may be born with that. Or it may be as a result of people like myself, because we are becoming older. So the lens loses its elasticity. So it remains more flat. It doesn't become convex. As one ages, the ciliary muscles are unable to contract enough. So one is the ciliary muscle, and one is that the lens loses its elasticity to become rounder. How can a person with nearsighted vision correct his or her vision? That's obvious. Glasses and contact lenses that are concave, biconcave, are prescribed to reduce refraction. Or the person can have surgery as well. Remember, laser surgery to adjust the shape of the cornea so that we can change and solve that problem. Remember, you must also know about cataracts, and you must also know about astigmatism. We must also know the pupillary mechanism. We have dealt with here near and far, so we have dealt with accommodation. Remember, accommodation deals with the lens, the ciliary muscles, and the suspensory ligaments. Those are the more important things. Pupillary mechanism deals with the iris and pupil. The pupil is just the space. Remember, the iris is the structure. It has two muscles, circular and radial. Don't confuse circular muscles for pupillary mechanisms with ciliary muscles, which is there for accommodation. Remember that all the time. And for pupillary mechanism, go back to old notes, and you'll see I taught you BCC and DRC. Bright light, circular muscles contract. Dim lights, radial muscles contract. And if the circular muscles contract, the radials will relax. And if the radials will contract, the circulars will relax. And the pupil becomes constricted or dilated according. Again, learn one, the other one is the opposite. Any questions so far? We take after the break.
Yeah, I think we'll
Okay,
have to
you
take
keep
them
it
after
ready
the
for us. Study the diagram below, still on nervous system, or rather sense organs, below, and answer the questions that follow. So they're giving you a diagram of the ear, and they ask you, first of all, in this diagram, which part has the receptors for balance? So you have to look for a labeled part here. There are two parts in the ear that are involved with balance. That is the semicircular canals and the utriculus and sacculus, which we call the vestibular apparatus. In this particular diagram, only the semicircular canals are labeled. That's D. So our answer would be D here. I'll come back to this balance issue just now. Transmits impulses to the brain. Which part transmits impulses to the brain? You know it's the auditory nerve. Look for the diagram. There's the auditory nerve there. So that is E. Equalizes the pressure on either side of part B. Part B being this part here. We'll talk about that just now. Equalizes, not balance and equilibrium, equalizes pressure on either side. And that would be this part here, known as the eustachian tube, and that is G. Amplify vibrations to the oval window. That will obviously be the ossicles, all of them together, and here we can only label C. In that case there. Okay, coming to hearing, people, look at this diagram and let's talk about how we hear. When you are talking about how we hear, all you do is follow the diagram. Sound waves are trapped by the pinna. So I'm going to give them numbers so you follow what I'm saying. So that's 1. That's the first part. 2 will be that sound waves go into the eustachian tube. 3 is it causes the tympanic membrane. 3. To vibrate. Notice we talked about sound waves first, now we're talking about vibration. That causes hammer, anvil, stirrup to vibrate. You're following the diagram, you're simply labeling the diagram when you're hearing. And that causes the oval window to vibrate. Vibration, second part. From there, this vibration go to the inner ear. They're going to the cochlea now. That is number 5 actually. This is 4, that will be 5. What happens in the cochlea? All you have to know is that the pressure waves, the stimulus, which is the pressure wave, is converted to an impulse by the organ of Corti, which is here in the cochlea. And there, from there, it goes into the auditory nerve. From the auditory nerve, it goes to the cerebrum, where it will be interpreted. How easy can it not be? You are just following a diagram to understand. Remember, in this last part, you're only talking about the organ of Corti converts a stimulus to an impulse to the auditory nerve to the brain where it's interpreted. Lloyd.
Well, guys, I hope you guys are keeping up with all the information coming at you. I hope you guys are going to go stretch your legs now. I hope you guys have got all the notes down. If you have any questions, remember you can go onto our Facebook page. We've also got a help desk that you can go to, which is learn.mindset.co.za/helpdesk. But guys, don't go away. We'll be back right after the break. Welcome back, Matric Life Science Mindsetters. Guys, I hope you guys have been keeping up with today's lesson. It's been really quick. It's been really action-packed, lots of information. Guys, we are going to try and answer as many questions as possible. There are thousands of questions coming through. And if we can't get to them in time, we will try and answer them at the end of the show. And if we don't answer them there's still the help desk you can go to. But for now, let's answer one question with regards to the ear. Aslam, there's a question that says, can you please explain how the semicircular canals are responsible for balance? And that's from Israel.
Israel, let's talk to you about that. Remember, the two things that are involved in balance are the semicircular canals there and the sacculus and the utriculus. Those are the two things that are involved in balance. Don't confuse balance with equal pressure. Eustachian tube. Balance means, and you'll understand what we mean by balance now when we explain. So one is you asking for the semicircular canals. The semicircular canals have cristae, or crista, plural cristae. This is found in the ampulla. Where's the ampulla? In the base of the semicircular canals. What does this thing do? They are sensitive. See how easy this is. They are sensitive to direction and speed of movement. In other words, if I'm walking forward, backwards, sideways, slow, fast. That's where it's all happening. You don't need to explain how it happens. You just need to know that this is a receptor that converts this stimulus of speed and movement into an impulse. And we're going to talk about the impulse right at the end. Then we have the saculus and the utriculus. They have maculae. S-U-M. Saculus, utriculus have maculae, some. And they tell us about the position. So you can make a little mnemonic there, thanks to Hamida there, SUMP. S-U-M-P. Saculus, utriculus have macula that help us with position of the head in relation to the body. That means at any given time, the brain knows that now my head is upright, now it's sideways, and this way, that way. Remember, when we taught this section, I did the headstand for you. Maybe, if you're lucky, I'll do that again today at the end of the session. Today, we don't have time now. So this is all you need to know. But now, the impulse, both of them, the maculae convert that stimulus into an impulse. This impulse goes to the vestibular branch of the auditory nerve. And that takes the message to the cerebellum. Remember, why the cerebellum? Because the cerebellum's function is balance and equilibrium. So whereas the auditory nerve for hearing goes to the cerebrum, because that's a sensation, perceived sensation, cerebrum. This one here, the vestibular branch, take it to the cerebellum, which will then instruct muscles, etc., accordingly on what to do. There's more complicated than what we are saying here. But for your level, this is all you need to know. I'm hoping that answered your question. Let's move on to your favorite section, and that is reproduction. The diagram below shows the human male and female reproductive systems. First of all, it tells you, write down the letter A to H and name of the part. That's a straightforward one. To do what? Which transports urine to outside of the body. In that particular case, it would be, in this case, it would be D, and the line should be coming closer there, should be the urethra. Where fertilization occurs, obviously fertilization takes place in a female, so you go onto this side, and that will be there, and the answer is E, the fallopian tube. You must know the structures of the male and female reproductive. I mean, you're all boys and girls, at least you should know that. Where sperms are produced, that would be the C, the testis or testes. C, one is a testis, two testes. That's not one test and two testes. Okay, where ova are produced, obviously ova eggs in the female, ovary, so that is F. Simple, instead of asking you the functions, we're simply asking this here, we're giving you the functions, and that is the ovary. Please look at the instruction. It says letter and name. Half of you leave half out, and that's not a good thing because you're going to lose half the marks, and you may need that half mark. Not half mark, but half the marks. That breaks down when the levels of progesterone and estrogen drop. That is obviously in the female, and that will be this part here, the endometrium G. Remember, that's not a very correct one, but in a bigger diagram it will be clearer. Endometrium. Where the Graafian follicles develop, that has to be the ovary, it will be again F, the ovary. So this is just dealing with structure and function. You also have to know the suitability of the different reproductive systems as well. It's obvious. You need to know that. And you need to know the functions of all the other parts, the seminal vesicles, the vas deferens, the epididymis, all of that. So this was just put for you to revise that. I'm going to skip this one and come back to this one. I'm going to this question here from Free State, June 2014. The following two graphs show the changes in temperature in a woman's body, as well as the levels of the hormones estrogen and progesterone during the menstrual cycle. Remember, so they're giving you the change in body temperature on the top graph, and the bottom graph is giving you what's happening between estrogen and progesterone. This graph is not talking about FSH and LH. Remember, whenever you're looking at a graph like this, or you're dealing with the menstrual cycle, keep FOLP at the back of your mind. FSH, estrogen, LH, and progesterone. Why FOLP? Because this is the order in which these hormones are secreted. If they didn't tell you this is estrogen and progesterone, you could use that to figure this out. That estrogen must rise before progesterone. Okay? So we have this situation there. We got that whole story there. You have it in front of you. Calculate by how many degrees Celsius the woman's body temperature varied in one menstrual cycle. So you've got to look at the beginning of the menstrual cycle and look at where it's going to. So you're going to take that, and you're going to just minus that, and you will work that out. It's straightforward calculation. 37.1 is the highest. 36.2 is the lowest. 0.9 degrees Celsius. Generally, you would have got a mark for, well, here you would have got a mark for that, and there are 2 marks. If this was a 3-mark question, you would have got 1 mark per unit. Always put the unit. It's not going to cost you anything, but it can get you a mark sometimes, okay, depending on the examiner and how that memo is described. From the diagrams representing the developing follicle, it appears that ovulation took place on day 14. From the graphs, list 2 more pieces of evidence which indicated that ovulation has occurred. Okay? So they're telling you there that what other evidence tells you that ovulation has taken place. One is that you can see in this graph, there's the formation of corpus luteum. Corpus luteum only forms when the Graafian follicle releases the egg. And you can see the egg coming out there anyway. And other than that, look at the time coincidence here. Look at that time coincidence there. Okay? So at the same time that this thing is telling you that ovulation is taking place, the temperature of this woman went up. That is generally when ovulation takes place. That's the time to see whether the person is fertile or not. But remember, it cannot be a reliable method of contraception because temperature can fluctuate because of other differences as well. Okay, so that's the story there. The temperature increased, the estrogen level decreased after ovulation. We also have to see there, obviously, that the progesterone level started increasing there as well. You can see that in the graph. Sorry, I didn't mention that. Did fertilization take place within the 28-day cycle? We're going back. When you want to see this, 2 things that you see. You look at the corpus luteum and you look at progesterone. If you want to know if it happens in that cycle. Most importantly, look at progesterone first. What's happening to progesterone here? It's going down. If progesterone is going down, it means fertilization didn't take place. Because if fertilization takes place, progesterone has to stay up because other than thickening the endometrium lining, progesterone also serves to maintain pregnancy. So if it's going down, that means pregnancy did not take place. Also, the corpus luteum degenerated very quickly here. It's decreasing in size very quickly in the cycle. So those are your 2 evidences that are there. I think I answered 2 questions in one. Yes, I did. Okay, so one is it's no. And give a reason. Progesterone levels decrease towards the end of the cycle is the main reason. Also, the corpus luteum degenerates quickly in the cycle. Describe the negative feedback mechanism involving progesterone and FSH in controlling the production of ova. The bottom line here is that you should know that when progesterone levels are high, FSH is low. When progesterone levels drop, FSH rises. This is the negative feedback mechanism. But we need to be able to explain why that takes place. So let's do that. First of all, this part here, this should have gone to the other answer, right? I told you that before. This one here, for some reason, little gremlin should go there on top. So that's not part of our answer here. Let's underline, draw that line. Otherwise, you're going to start writing that in your exams. Increasing levels of progesterone secreted by the corpus luteum inhibit the release of FSH from the hypophysis or the pituitary gland. This prevents. Why does this happen? Why? Because we're explaining. Explaining means you must say what happens and you must say why it happens. This prevents the development of any other follicles as well as ova. Remember what's happening. A follicle is already in the stages of development if progesterone is high, or the woman is pregnant if progesterone is high. We do not need another follicle to develop. Why is this so? Because we already got one cake in the oven, we don't want another cake to come up there, okay? To put it crudely, Lloyd. Huh? Okay, I can hear Belinda laughing in the background there as well. I hope so, anyway. So what we're saying here is that, remember, why am I saying that? Remember, a woman, the number of eggs she will develop in her lifetime is determined when she's born. So she has stock of X amount of eggs. Now, if there's one egg that is already traveling in the fallopian tube, whether its fertilization has taken place or not important, but it is moving through the process. We don't want another egg to go up there as well, because it would be wasteful. Okay? So that is why this process is in place there. If the now the other part, remember we explained only if progesterone levels are high. When progesterone levels drop, if fertilization does not take place, then the secretion of FSH is no longer inhibited. That means FSH will be stimulated again and the new cycle continues. That means after the cycle is over, the next cycle starts, the next follicle will develop and so on. So that's what we are talking about. Very important to explain this relationship between these two things. Okay, so that's reproduction. Remember again, in reproduction, you need to know all of that, and you need to know all the hormones and their functions. You also need to know, in terms of fertility, etc. You need to know about infertility a little bit and so on. You need to be able to talk about that. Generally with data response questions and so on, you can get questions on that. So you need to know the story in terms of that as well. Also about what happens to the ovum. The ovum becomes a zygote when fertilization takes place. This zygote becomes the morula. The morula becomes the blastocyst for our study purposes. There are other stages that we can talk about, and that will become implanted. Eventually, after 3 months, we call it the fetus. That will be followed by birth after the pregnancy, gestation, and then birth. Okay, so that's what you need to know there. Let's move on to homeostasis question. We'll do this one quickly. Answer the following question based on the graph on body temperature below. So what they're telling you here, there's time, and something is happening, temperature is going up, then it's coming down, then it's going up again. And this is the norm. Okay, so you need to read that. Describe the changes that will take place in the skin to reduce the temperature from point X to point Y. That means coming down there to where it is here in this area. But actually, I would change the question from point X around there to normal. In other words, another way of asking this question, what happens when the temperature of the body rises above normal? You must be able to read that in this situation. Now, this is a simple question, and we're going to just go through that quickly. First of all, remember that they're only asking for the reaction as such. But remember, in any of these questions, I would advise you to look at detection first, control center, and response. So here we're going to say that the receptors detect the stimulus. They send that to the control center, which is the hypothalamus. So that's the first part of the answer. Then you go to our answer, the response. The blood vessels dilate, vasodilation. Which blood vessels? Of the skin. Dilate or vasodilation takes place. More blood flows to the surface. More heat goes to the surface and therefore is lost. More blood flows to the sweat glands. Sweat glands release more sweat. More evaporation and more cooling of the skin takes place to bring the temperature back to normal. Lloyd.
So guys, before we go to our ad break, remember about our K53 Safeways competition where you could walk away with a brand new Chevrolet Spark L, the first one of which will be given away on Sunday at 5 p.m. Remember to tune in. But guys, before we carry on, let's take a quick break. Don't go away. Welcome back, Matric Mindsetters, to today's Life Science Exam School. I hope you guys had a quick break, stretched your legs, and you're ready to learn some more. Aslam, should we get straight into it?
Kick straight into it. Thanks, Lloyd. I like that. I like that. We don't have much time, so we talk less and we do more. Yeah. Okay. Welcome back, Mindsetters. So we were talking about what happens in the skin on a hot day. Remember, your focus is on what happens to the blood vessels, and that can either be constrict or dilate, and what happens to the sweat gland. Sweat more or sweat less. Now remember, C for cold, C for constrict. So if you remember that, then already your bulk of your work is done. And if on a hot day, dilation is taking place. So on a cold day, what's going to happen? C for cold, C for constrict. So that's what you're going to say there. You're going to talk about constriction, or you're going to talk about vasoconstriction. And all the mores become less. That's all that changes here. Look at that. How many times? So you're changing two words, Lloyd. You're changing dilation to constriction. And you're changing all the mores to less. In that way, you got the answer for a cold day as well. It's how easy it can be. And yet, look at here, even it becomes less. Less sweat. Less. Look at that. Less. You only change one word, more to less. And that's what you're going to get on the other side. So please, don't learn both things, learn one of those things. By the way, we'll talk about the amounts that I gave you earlier. I just want to correct something I've made a mistake with, especially on reproduction, maybe because that's my favorite section. I give it extra marks, but when we go back, we'll show you in the last session. Where is the temporal regulatory center located? We spoke about it already, and that is the hypothalamus. Study the diagram of the cross-section to the blood vessels in the skin under different conditions. There's A and B. Look at the diameter of these blood vessels, this one and that one there. That's the clue. Which diagram best shows the state of the blood vessel at position Y in the graph on page 4.1? So position Y is lower temperature, obviously. So obviously, that will be then will be that particular one there. A will be the one where the vasoconstriction is shown there. It's narrower in diameter. Justify your choice. It's the same as we've already explained on the top. At Y, body temperature is below normal, so vasoconstriction occurs until, or rather to limit heat loss from the surface, and also so sweat glands less stimulated. I just want to just highlight one other thing. When you're describing this situation on a hot or a cold day, always explain what happens to the blood vessels first, so it makes more sense. When you're talking about the blood vessels, when they are constricted, then when you move to the next part, then you say that less blood goes via the sweat glands, so there's less sweat produced. So you are linking the whole thing up. It makes more logical sense that way. Okay, so that will cover you there. Let's move on. Notice for the exam guideline, we don't talk about the hair and erector muscles. We are focusing mainly on the blood vessels and the sweat glands. Right, here, Anela found that she was always tired, weak and tired. She was concerned, so she went to the doctor who did some blood tests. The blood tests show that her TSH levels were 0.02 micrograms per deciliter. The normal range for this hormone is 0.4 to 4.5. So obviously, 0.02. And the normal range is 0.4. So obviously, she's got a problem. Name the gland that secretes TSH. Now, remember, earlier we did homeostasis. Now we are touching on endocrine system and a little bit of homeostasis as well. Name the gland that secretes TSH. It will be the hypophysis. Explain why this gland is an endocrine gland, and that means you're defining an endocrine gland. When a gland secretes its secretion directly into the blood, it's known as endocrine. When it secretes its secretion into a duct, which takes it to where it must go, then it's an exocrine gland. So that's our answer there for that one there. It's secretion directly into the blood. Explain why the low levels of TSH causes the symptoms that Anele experienced. Let's go back to the symptoms. She was tired and weak. Tired and weak. Right? So why is that so? TSH, let's name TSH first of all. TSH is thyroid stimulating hormone. So it stimulates the thyroid gland. So obviously if the TSH is low, then is an underactive thyroid gland. And what's the function of the thyroid gland? To secrete thyroxine. What is the function of thyroxine? Is to regulate the metabolic rate. So if TSH is low, thyroxine is going to be low, and because of that, the metabolic rate will be low. What does that mean? That means we are burning food, our respiration rate is slow compared to normal, and therefore we are not getting the maximum energy from the food we eat. It can also lead to weight increase and so on and so on and so on. This question is actually linking negative feedback because you're asking about TSH and you're rippling back to thyroid gland as well. So you need to know that. Okay, so TSH stimulates the thyroid gland to secrete thyroxine. Thyroxine controls the body metabolism. Low levels of TSH, low levels of thyroxine. And this is a low metabolism, and the person feels tired because there's not enough energy, right? Underactive. Next question, high levels of TSH. High levels of TSH in the body increases the heart rate. Name one other hormone produced by the body that will have the same effect. That's a simple question. It would be adrenaline. Okay? So we're testing on that. Just remember, by the way, in homeostasis, in addition to what we have done, we've only done thermal regulation. In homeostasis, you also have to know, you look at your trial papers, you'll see they tested you on osmoregulation as well. They can test you on the carbon dioxide concentration, and they can test you on the salt concentration, and they can also test you on glucose. Remember, glucose, you're talking about insulin and glucagon. Glucagon. So you need to know these things as well in homeostasis. And in the endocrine system, you need to know all the glands, the hormones they secrete, and the functions of those hormones. For insulin, you need to know under secretion of insulin what can happen, and for thyroxine, the same as well. Question 5. Scientists at the drug company developed a new pain-killing drug called X. The scientists compared the drug with 2 other pain-killing drugs, A and B. In their investigation, the scientists chose 600 volunteers. All of them were in pain. Gave 200 volunteers a standard dose of drug A, B to the other 200, and 200 had X. Over the next 7 hours, the volunteers recorded how much of pain they felt. To get valid results, the scientists kept as many factors constant with regards to the 3 groups as was possible. The graph below shows the results of this investigation. So, on the x-axis, time after taking the drug. Here, the decreased percentage pain. Mean percentage means they took the average. Okay? So they're giving you the story here. Drug A, amount of pain. Drug B and drug X. That's the graph that we have. Okay? Which part of the brain is responsible for the interpretation of pain stimuli? Sensations, obviously the cerebrum. Which drug A, B, or X was most effective? Again, I'm going to rush to that X, but let's go back to the graph. And there, remember, very careful to look at here, mean percentage decrease in pain. That means the higher up it is, the less pain. So don't be confused, because some people may look at what's happening here at the end here and going down. But in the meanwhile, they're saying decrease in pain. The higher, that means the higher percentage of the pain decrease. So it'll be drug X, obviously. Least effective in the first 2 hours would be drug B, and you can look at that on the graph. How long did it take the drug named in question 5.28 to reach maximum effectiveness? Again, 2 hours, 18 minutes. You have to read that from the graph. Let's go back there. Maximum effectiveness would be around this area here. So you're going to read across here, and you're going to work out, because you got grids here, you can work out the time. Obviously, there will be a small range available there. So you're reading, you're going down with time. This is the area. You're going to look at the highest point where it reached. I think it's there, going right down there and coming down, and you're going to work that out. Why is that 2.18 minutes there? Okay? State why the scientists used 600 volunteers instead of only 100. Obvious. Look at the numbers. more volunteers, increasing the sample size. So therefore, is to make the reliability of this investigation better, to be more reliable. So increase sample size, reliability, increase the sample size, or repeat the investigation several times. What is reliable? Means that we can trust the results. How can we trust it? If every time we do it, we're getting more or less the same result. That's why we need to do it more than once. We can't do it once only because that could be a fluke. List 2 factors that would have been kept constant between the groups to ensure valid results. Remember, when we're talking about keeping constant, when we're talking about correct method, then we are talking about validity. When we're talking about the results being trustworthy, we're talking about reliability. Okay? And constant in this case, we have to keep the people that we're using at the age must be the same, obviously. We can't have a 5-year-old and a 25-year-old and a 105-year-old and expect the same results. The gender, because different people, different genders have different pain thresholds. The general health, if someone is more sick than the other person, then obviously the pain thresholds would be different. Not on any other medication. They mustn't be taking other medications at that time. Quantity of each drug administered. So in other words, each one must, if we're giving A, B, and C different drugs, we're giving the one 500 milligrams, we must give all 500 milligrams of the drug that is A, B, or C. Okay? Those are some of the things that must be kept constant. Question 6. Here's a story now on the plant hormones. Thabo did an investigation in a school lab to study the effect of different concentrations of auxins on cell elongation in coleoptiles, in young stems. He used the following procedure, 15 coleoptiles, all were used of the same length. Notice the constant. The tips of 12 coleoptiles were removed. Right? These coleoptiles were put into 4 groups, A, B, C, and D. Each group of the 3 coleoptiles was injected with a different concentration of auxins, as shown in the table below. Notice when you get an investigation question, people, this is the plan that you'll always take. There's a little opener there telling you about what this investigation, and generally the aim will be found in this first part. Thereafter, what the person has done is listed in bullets. It'll always be like this so that it's easier for you to read. The last group E was used as a control in which the coleoptiles were not injected with auxins and the tips were not removed. So you got all these things here, tip present or removed, absent, 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 present. Okay? Concentration of auxins, 1, 2, 3, 4, and there's the average length there. Formulate a hypothesis for this investigation. Now look, we look at investigation. We looked at dependent and independent variables earlier. We looked at reliability. We looked at validity. Now we are looking at a hypothesis. A hypothesis is a prediction. You can't make it a question. You can't make it an aim. You can't make it a conclusion. Ideally, a hypothesis is in the future tense, or in the present tense also, because normally a hypothesis is done before we do the investigation. But we can't do that here. We have to give you what we're talking about to ask the question. So the hypothesis in this case would be cell elongation in the coleoptiles will increase as the auxin concentration increases. One hypothesis. Or the elongation will decrease with the increased auxin, or with a decrease, etc. Or they remain the same, or the auxin concentration will have no effect on the length or the elongation of the coleoptiles. Notice we're not saying, will the coleoptiles increase if auxin concentration increase? We're not asking a question. We're not saying to determine if, that's an aim. Okay? So keep that in mind when you're answering that. Suggest the reason why Thabo had to remove the tips of each coleoptile before injecting them with auxins. That's straightforward. To remove the effect of auxin that is produced by the tip itself. Remember, the growing tip is where auxin is produced. So by taking away the tip, we're taking away the auxin. So we are now determining how much auxin in each of those things there. And that's important for us, because that's what we're investigating. What conclusion can be drawn from the results about the effect of Thus, it's obvious that an increase in the concentration of auxins resulted in the increase in cell elongation up to an optimum concentration, and thereafter it was inhibited or decreased the cell elongation. And with that, Lloyd.
So guys, I hope that's really summarized a lot of things in your head for this session. But guys, before we go, remember about our Safeway K53 competition. You could be that lucky winner of that Chevrolet Spark Light. And it could be you. All you had to do is purchase one of these books. And you won't be in this draw coming up on Sunday, but there are still another 3 cars to give away, guys. So get your K53 book, enter on our Facebook page, and it could be you. But don't go away. We'll be right back after the break. Welcome back, Life Science Mindsetters. Before we carry on, I hope you guys have had a look at our K53 Safeway K53 competition where you could win yourself a Chevrolet Spark L. And guys, please remember that today's show is sponsored by the MCN SA Foundation and is endorsed by the Department of Basic Education. So without further ado, let's carry on.
Thank you, Lloyd. Right. So remember, this section dealt with auxins. Although it was experimental, investigative, remember you need to know the functions of the hormones, that means auxins, abscisic acid, ABA, and gibberellins. You need to know their functions. Auxins, you must also know apical dominance, in more detail, phototropism, and geotropism. You must be able to explain those concepts as well. And in terms of hormones, you must also know about plant, or rather not on that, on plants, you must know plants' defense mechanisms, how plants protect themselves. So that covers that section. We just tip of the iceberg. We were just looking at a little bit on the functions of auxins. Lloyd, you had some interesting questions there, I noticed during the break.
Yeah,
Can
guys.
we take
so
the first one.
We've picked out a few of your guys' questions, and let's take a look at those. The first question is, please explain why the continued production of adrenaline can lead to the high levels of lactic acid in the muscles. And that's from Rendani.
Excellent question. It's not really prescribed in the syllabus, but I like the way you put that question. Why is there, if there's a prolonged increase in adrenaline, we have lactic acid? Function of adrenaline is to prepare for emergency. And one of that is to send blood, more blood to the muscles, skeletal muscles. And the skeletal muscles, if they're going to get more of that blood, then obviously their metabolic rate may be higher. And if there's not enough oxygen being supplied at the same rate, then obviously anaerobic respiration will take place, and therefore lactic acid will result. Remember, lactic acid fermentation is not prescribed in the syllabus, so it's not necessary to know the story there, but that's the story that I'm giving you just so that you can, it's interesting to pick it up. Next.
Yeah. Okay, guys, so the next question is from Tlengiwe, and Tlengiwe says, hey, Aslam, can you please elaborate on the birth process, labor, expulsion, etc.?
Shlengiwe, you know, if I was capable of giving birth, I would have expanded, I would have labored at that point, but I can't do that. But in terms of birth, you only need to know a couple of things there. One is the breaking of the waters.
So just to clarify, this birth process is in paper 1.
It is in paper 1, yeah.
Okay.
Reproduction, right? So breaking of the water, that means the amniotic membrane, the amnion breaking and releasing the amniotic fluid. That's the first part. And this will be followed by labor. That means contractions of the muscles of the abdomen and the uterus. And this is followed by expulsion of the baby. That's all you need to know. You don't have to go into details about the hormones involved, nothing. You're simply going to say that these are the 3 stages in the birth process. Next one, the last one.
Okay, guys, and the last question that we've picked out is, can you explain why, if the corpus luteum remains after fertilization has occurred, no new follicles will be formed?
Come, guys, we answered that question earlier. If the corpus luteum is there, what does it secrete? It secretes progesterone. Yes? And we said earlier, if progesterone levels are high, it inhibits FSH. What is FSH again? Follicle stimulating hormone. So progesterone is high, that means no FSH, and if there's no follicle stimulating hormone, there will be no more follicles. And why does it happen? I told you earlier that we don't want unnecessary wastage of follicles. That's it?
Yeah?
Okay, let's move on then. Okay, so we've covered that, but let's move on to a essay type of question. And this was taken from the prep, KZN prep paper. A snake slithers over Sifiso's foot. He immediately jumps away from the snake in fear and manages to maintain his balance as it slithers. Describe all the events that lead to Sifiso's reaction. Now, this is a straightforward question on, one is on reflex action and two is on balance. So you need to pick that up from there. So where's the verb? Describe. Whatever comes after the verb is what you must do. All the events. You must describe all the events. What did he do? He jumped away from that and he maintained his balance. Those are the two things that you needed to explain here. And I'm just going to open up the answer. We're not going to waste too much of time with that. Okay, so first we are increasing all the stories there about the fact that he got scared. There's a bit of adrenaline there. That's one. Two is we got the balance in that way as well. And we can also add a little bit of reflex action in that because he pulled away his leg quickly there. Describe how poor I'm sorry if I'm rushing through the essay. We don't have time to go through the essay questions. It's just to expose you on the skill of writing the essay. We're not going to discuss the entire essay. Describe how poor farming practices may impact negatively on water quality, water availability, food security, and biodiversity. This is a huge essay with a huge potential of plenty marks. We could have even broken it up to here. And that could have made an essay. Or these two, they could have made an essay as well. So it could be the whole thing, or this was taken from Northwest Paper Prep 1. And the potential answer that we're looking at there is what happens there, all pest control causes, and this is on your page so you can go through it at leisure. Now we come to the challenge question. Lloyd, while we're doing that, you can just do a bit of a scan if you see some other interesting questions.
Yeah.
If we have some time in the end. Because I've left one question out, I'm going to go back to that as well. Okay. So here let's look at this one first. We have to look at the diagram. Identify the process of the yes, it is fertilization. Okay, we know that. Where in the human body does this take place? We answer that with the diagram. Has to be in the fallopian tube, generally. Assume structure B had 24 chromosomes. What is structure B? That is the nucleus of the egg. Okay? Explain how structure B. Obvious, this is asking you for non-disjunction. It's another way of asking you non-disjunction. I like that. That's why I put it there. And the homologous chromosome pair does not separate, or non-disjunction. Non-disjunction means does not separate during anaphase 1 of meiosis. And because of that, it's only one we're talking about, one homologous chromosome pair that did not separate. Hence, the person has 24 instead of 23. How many chromosomes would structure C have if structure A was normal? What is structure C? Structure C is the result of the fusion of the sperm and the egg, so it is a zygote. 24 plus 23, that's just a simple arithmetic, will give you 47. How many chromosomes would structure C have if A was normal? Astam, you got that one wrong completely. So it would be 47, because this one has, is the zygote, it's A plus B, or whatever the letter was. If A was normal, it's 23, and B is 24. So the two together will give you 47 because the person has one extra chromosome. I'm hoping that makes sense. Now remember, we can ask the same question in Down syndrome. Generally, more asked in paper 2 because meiosis proper is tested more in paper 2. However, meiosis as a total is prescribed in both papers, so you need to know that. I just want to go to the CAPS document to correct what I said earlier. Remember, we said meiosis was 11 marks. Human reproduction in humans, I said 41, it's 31 marks. Okay, 6 marks for vertebrates. Responding to the environment is 40, that was right. Endocrine is 15, that was right. Homeostasis 11, and 11 for plants, hormones, and 25 marks for human impact. That's your paper 1. This is the guideline in terms of mark description. But more important is you must know these topics. These topics are in the paper, so you need to know those topics. Keep that in mind. Okay, so we corrected that. I just was more generous with reproduction. I won't say why. I said I left one question out, and that's this one here. I say, when men and women are unable to have children, we say that they are infertile. There are many reasons for infertility, and the pie chart gives you all the reasons for that. Sperm problems, failure to ovulate, damaged fallopian tubes, etc. You've got the graph so you can look at it. Sperm problems in males may occur as a result of deformed sperm. These sperm may have very short or no tails at all. They have very short or no tails at all. Explain why this deformity. That's easy, guys. If it doesn't have the tail, it can't swim, it won't reach the egg in time. That's the general crux of the thing there. With a short tail, the sperm would not be able to swim, they would not be able to reach the ovum, and therefore no fertilization can take place. Name the parts of the male reproductive system where sperm cells are produced. It's obvious, we've answered that before. The testes, where they mature, that will be the epididymis. Remember, from the testes, the sperms move to the epididymis, where they are stored for a while and they mature. From there, they go to the vas deferens, and then they go right down to the ejaculatory duct. While they're going to the ejaculatory duct, substances from seminal vesicles, the Cowper's glands, and the prostate glands are added to the sperm, and from the ejaculatory duct, they will be released via the urethra. Remember, those 3 glands, they add fluid, they add food, nourishment, and they add alkalinity to the sperm. You can look at the specific functions there. 14% of all the cases of infertility that are reported are a result of damaged fallopian tubes. Explain how damage to the fallopian tubes may cause infertility. That's also not too difficult, straightforward. The damaged fallopian tubes could be blocked, and if they block, that means the sperm are unable to reach the egg to fertilize it, or if fertilization takes place, the zygote may not be able to pass down fallopian tube to reach the uterus. Failure to ovulate may be treated artificially by supplying hormones to increase the chances of ovulation occur. What percentage of people could benefit from this type? Again, you got the graph. I'm just opening it up there. 18. Name one hormone that may be given to a woman who is not ovulating and explain why this hormone is of benefit. That's a nice question. It should be follicle stimulating hormone to stimulate the development of the follicle, or luteinizing hormone which can stimulate ovulation. Name the structure of the female after fertilization, and that we already spoke about that earlier, and that is a zygote. It divides to form a morula, which further divides to form a blastula, or blastocyst, eventually a hollow ball of cells. Any questions?
There's one that's gotten my attention. Why does the ovary not consist of long convoluted tubes like the testes?
The ovary has already the number of follicles that are going to be in the woman's life. Whereas in the seminiferous tubules, sperms are being produced all the time. So they increase the surface area for the production of sperm. Whereas in the female's ovary, the number of eggs, the stock that she has, is already in the shelf, if we want to put it that way. And this stock is going to be released from time to time. So there's no need to increase the surface area for that to happen. Any other questions?
Just a quick one. What is implantation?
What is implantation, somebody asked, is when the fertilized egg, the morula, the blastocyst, eventually becomes attached to the endometrium wall. We call this implantation.
Cool, guys. I hope that's answered all the questions that you had. If we haven't, you can still go onto our help desk. That's at learn.mindset.co.za/helpdesk. Thanks a lot, Aslam, for your help.
All the best, Matrix.
Yeah, guys, we've had a lot of compliments for Aslam, so thanks a lot for teaching us, Aslam. Guys, if you still need any help, you can go over onto our help desk. Also, all the pages from today's lesson will be on Facebook, and you can still enter into our Safeway Case 53. But guys, until next time, I hope you guys keep well. Good luck with your exams, and we'll see you next time.